Hi friends, welcome back to WIP's Biochemistry, and this time it's electron transport chain and oxidative phosphorylation. This is the part 1, and this is actually continuation of cellular respiration. And what is cellular respiration? It is basically generation of ATP with oxygen being used as the ultimate electron acceptor. And the oxidative degradation of food molecules like amino acids, fats, or carbohydrates, they end up in electron transport chain or converge in electron transport chain and oxidative phosphorylation. So actually the significance of this process is that an average person requires around 2800 kilocalories of energy or kilocalories of ATP. This actually amounts to 380 moles of ATP, which equal to 190 kilograms of ATP. I do mean kilograms, 190 kilograms of ATP per day. And whereas a mere 50 grams will be available at a point of time in the body of an average person. So that means we require a lot of ATP per day. And that also means that thousands of times ATP will be broken down to ADP and will be resynthesized as ATP. So that is the story of electron transport chain and oxidative phosphorylation. So let us see. So first of all, we can have a quick recap of the processes which lead to electron transport and oxidative phosphorylation. This picture shown here is an exclusive one which shows the substrates for electron transport as well as oxidative phosphorylation, which are fed from different sites by oxidative degradation of food molecules. So this is TCA cycle to which these molecules are fed, like this, molecules are fed, and this is the electron transport chain, the series through which the electrons pass through. You can split this picture and see. Now these molecules here, amino acids, pyruvate, fatty acids, these are actually formed from as a result of oxidative degradation of food molecules. And obviously, for example, pyruvate is formed from glucose through glycolysis, and acetyl coenzyme is here. So these are the molecules which are fed into TCA cycle. Now, the function of a TCA cycle or citric acid cycle is the harvesting of high energy electrons from these carbon fuels. And you have to note here that the citric acid cycle itself neither generates a large amount of ATP, does not produce significant quantities of ATP or GTP, nor it includes oxygen or uses oxygen, molecular oxygen as a reactant. So molecular oxygen is not directly involved as a reactant in TCA cycle. Instead, citric acid cycle removes electrons from acetyl coenzyme as well as some other substrates as shown here, and uses these electrons to form NADH as well as FADH2. So this is the basic purpose as far as in the last picture. This is the basic purpose of TCA cycle. Now we move on to here. This is electron transport and oxidative phosphorylation. The NADH, the electrons from NADH and FADH2 are passed through this electron transport chain. So that's how oxidative phosphorylation also occurs. We will see later. Oxidative phosphorylation is the process in which ATP is formed as a result of the transfer of electrons from NADH and as also FADH2 to molecular oxygen. So electrons are accepted finally by molecular oxygen, and water is formed. Now here there are 5 different types of cytochromes. You can identify 5 different types of cytochromes here between this point, ubiquinone, and this particular reaction. There are 5 different types of cytochromes, but they are termed or named based on the wavelength of light they absorb, which unfortunately it is not in the order in which they appear here. You can see the order, this B first, then C1, cytochrome C1, then cytochrome C, then cytochrome A, and cytochrome A3. So this is the order in which they are arranged in the mitochondrial membrane. So this is actually the nutshell of the entire process which we are going to see in detail. Now we cannot proceed with explaining electron transport chain as well as the process of oxidative phosphorylation without explaining without having proper understanding of the structure of mitochondria. Now, a mitochondrion is shown here with the outer membrane and an inner membrane as shown, and in between there is an intermembrane space. So these membranes and the space are important, as well as matrix where Krebs cycle occur. So that is important. And apart from that, there is cristae, which are invaginations to the matrix, that is the invagination of inner membrane to the matrix, as well as on this cristae we can see what is called FOF1 particles, which are also called ATP synthase molecules. So that we will see in detail later. Now, the matrix side is also called the N side, indicating it's having a negative charge, whereas the cytosolic side, that is the intermembrane space, it is having a positive charge, so it is also called the P side. Mitochondrion is the organelle where the final stage of cellular respiration occurs, and cells that use lots of energy have a higher number of mitochondria, for example heart muscle cells. Now, it is on this inner membrane that is the electron transport assemblies or complexes are arranged. So their arrangement can be seen in further slides. And a single the inner membrane of a single mitochondrion may have more than 10,000 sets of electron transport chains as well as ATP synthase molecules in it. Now, first of all, let us get familiarized with the names of the complexes, electron transport chain complexes or respiratory complexes. Here, the first one, complex 1, is NADH oxidoreductase. The second one is termed Succinate Q reductase. Then this one, third, complex 3 is Cytochrome C oxidoreductase, and this one is complex 4, Cytochrome C oxidase. And in between, there are two mobile carriers of electrons also. This is Ubiquinone and this is Cytochrome C. These are mobile carriers. It is actually through these complexes and carriers that electrons are passed, resulting in proton pumping, which we'll see, and also resulting in electrons. These electrons being passed or accepted by molecular oxygen so as to get converted to water or to form water. Let us see the process of electron transport, proton pumping, as well as formation of water or acceptance of electrons by molecular oxygen to form water. So these processes can be seen in detail here, and this is mitochondria showing respiratory assembly. This is the outer membrane of mitochondria, and this one is inner membrane, outer membrane, inner membrane, and this is the intermembrane space. So all these are having significances in the process of electron transport and this oxidative phosphorylation. And this part is mitochondrial matrix. Now, the molecules in electron transport chain are arranged as per their redox potential. This arrangement is as per their redox potential. That we will discuss in detail in coming slides. The electrons from NADH here, it passes to complex 1, but it actually passed to FMN prosthetic group, a tightly attached FMN prosthetic group of NADH dehydrogenase enzyme of complex 1. This enzyme is having 34 polypeptide chains, and this enzyme oxidizes NADH as shown here. So H plus is released, as well as the electrons are passed to like this. It is passed to FMN, and FMN gets reduced to FMNH2, and these electrons are passed to complex 1. So complex 1, as you can see, it's an inverted L-shaped structure with the base embedded in the inner membrane and the rest directed to the matrix, as you can see. And it actually consists of 46 polypeptides. Now, complex 1, it receives electrons from NADH here, and the captured electrons are passed through a series of FeS clusters, iron sulfur clusters, in complex 1. The iron is actually not part of heme structure, just like you see heme in hemoglobin or heme in cytochromes. This is not part of hemes. It is a non-heme iron molecule, and it is found normally attached to inorganic sulfur atoms, or sometimes to sulfur of cysteine or a sulfur of histidine in proteins. And mostly it is seen as Fe2S2 clusters or Fe4S4 clusters with inorganic atoms, inorganic sulfur atoms. And you can see here that FES clusters also occur in photosynthetic electron transport chain also. And those FES clusters are believed to be evolutionary precursor of this oxidative electron transport chain. And this is how electrons travel through complex 1. And as we have studied in physics, the flow of electrons is what creates electric current. And this energy is responsible for the expulsion of H+. You can see the H+ here. How many number of H+? 4 numbers of 4 H+ molecules are transferred or pumped. Actually, it is pumped. The term used is pumping of H+ or pumping of protons from mitochondrial matrix to intermembrane space. So this is a very much a significant process because it tend to accumulate H+ ions in the intermembrane space, and we can see in coming slides what is the significance of accumulating H+ here. Now this is what happens, and as a result the electrons in complex 1, as you can see, it gets the electrons get passed to ubiquinone. So this is ubiquinone shown here. Ubiquinone accepts electrons and get reduced to ubiquinol. And ubiquinone, it is formerly called coenzyme Q. It's not a protein, but are compounds having 6 to 10 isoprene units in them. Generally, mammals have 10 isoprene units in them. So it is called, in mammals, it is called Q10. And it is not covalently attached to any protein. It's just a mobile carrier of electrons. So when we say that FMN, flavin mononucleotide, gets reduced to FMNH2, what we mean is that by acceptance of electrons here, nitrogen here and here get reduced, so that it gets converted to FMNH2, and these electrons further pass to complex 1. And as well as ubiquinone accepts electrons from complex 1. That means like here, here O, double bond O gets reduced here, so to become ubiquinol. Now besides accepting electrons from complex 1, ubiquinone molecules actually accept electrons from complex 2 also. Now complex 2 is attached to the inner portion of the inner membrane of mitochondria, as you can see here, and complex 2 actually it does not produce sufficient amount of energy, so that it cannot pump out protons. This is the reason why it's not a proton pump. This one is unlike complex 1. Complex 2 is not a proton pump. Succinate dehydrogenase enzyme is a part of this enzyme, and you may recall that succinate dehydrogenase actually catalyzes the conversion of succinate to fumarate with release of FADH2 in one of the steps in T-cell cycle. Now, the FADH2 produced there, it remains attached to this complex 2, and passes these electrons to the iron sulfur cluster of complex 2. So this is a curious point, that this FADH2 still remain attached to complex 2 and passes its electrons to complex 2, and which eventually will be passed to ubiquinone, so as to reduce ubiquinone to ubiquinol. And further, these electrons from ubiquinol passes to complex 3, as you can see here. Complex 3 accepts these electrons, and complex 3 actually contains cytochrome C1 with a heme group, cytochrome B with the 2 heme groups actually, and ReSK center also. ReSK center is exclusive to complex 3. It contains 2 iron, 2 sulfur cluster groups in ReSK center. Now, function of cytochrome C oxidoreductase is to catalyze the transfer of electrons from ubiquinol to cytochrome C, the oxidized cytochrome C. Now, cytochrome C, as you can see here, is a water-soluble protein. It's a mobile carrier of electrons, and what happens is that it accepts electrons from complex 3, and as a result, the protons are pumped out of mitochondrial matrix. How many of them? 4 H plus molecules are pumped out of mitochondrial matrix towards intermembrane space. The entire travel of electrons from reduced ubiquinol to cytochrome C here, this mobile carrier, is called Q cycle, which end up in pumping of 4 H plus molecules to the intermembrane space. The final stage of the electron transport chain is the oxidation of the reduced cytochrome here. This is coupled to the reduction of oxygen to 2 molecules of water, as you can see here. And this is done by complex 4. In complex 4, actually a total of 4 cytochrome molecules are oxidized like this. This is reduced, and cytochrome C gets oxidized. The whole process is accompanied by expulsion of 2 H plus ions like here, and to the intermembrane spaces. The final oxidation is carried out with the help of oxygen itself, and it's a complex step in electron transport, since 4 electrons must be passed almost simultaneously to molecular oxygen like this. These two processes must be coupled to yield 2 water molecules with uptake of 4 H plus from matrix. So the H plus is taken from matrix, that too 4 of them. Here only cytochrome A3, which is part of cytochrome 4 in complex 4, can only react directly with the oxygen. And also note that any kind of partial transfer of electrons to oxygen will result in production of hydrogen peroxide or superoxide free radicals that can damage membranes and vital organs. So what is actually achieved here is that here is an accumulation of protons in intermembrane space and generation of a proton or pH gradient or an electrical gradient across this membrane. Otherwise, electron motive force is converted to proton motive force, and proton motive force is converted to phosphoryl transfer potential. So that is the role of this particular complex here, FOF1 or ATP synthase molecules, which is also called complex 5 here. And this generation of ATP using proton motive force is done by this FOF1 complex, and which is explained by chemiosmotic theory, and that we will discuss in a future lecture. This is a better illustration of the complexes. You can see the complex 1, 2, 3, 4 here, as well as ubiquinone and the mobile carrier cytochrome C are also shown. And this is the first acceptor of electron from NADH, FMN here. Then Q cycling is illustrated here, as well as the cytochrome B, C and iron sulfur clusters are shown here. You can just pause this figure and go into the detail. And here is the final cytochrome A3, which is responsible for the acceptance of electron by oxygen. So final oxidation to give water molecule.